In this video, we're going to be talking about the different kinds of genetic mapping, or the mechanisms that we've been using since the beginning of the 20th century to actually map the position of genes within chromosomes. So the first one is the one we talked about on the last video, which is the idea of linkage mapping, or that uses recombination frequencies between two different genes in order to position them within the chromosome. So for example, if you know that the recombination frequency between the appendages on the head and the color of the body is 48.5%, you know that they are this far in the chromosome. And then if you know that between the gray body and the color of the eyes is a little bit less than 9%, you know that you have to jump another 9% about that. But how do you know if that 9% is to the right or to the left of that? Because this 9% could fall somewhere here also, because this could also be 9%. Well, you know that's 9% because when you calculate this combination frequency, you get 57%, right? And then the same thing is true about, let's say, if you want to put normal wings with gray body, you know that this is going to be something close to whatever the difference between 65 and 48 is, so this could be something close to 17%, right? But you know, how do you know the red eye is supposed to fall on the bottom, in between them, and not somewhere down there? You know, because you know this recombination frequency and so forth. So in other words, when you look at all the recombination frequencies between all the traits which are supposedly within the same chromosome, you can create a map of the position of these traits within the chromosome, and that's what linkage mapping is all about. Remember that two genes which are close together have a very low chance of crossing over together, because two crossing over events are not really going to happen close to each other, because to form two chiasmata really close to each other, you have to bend the DNA too much, which will cause problems in the transposition events which are required during crossing over. And maybe you would end up with the genes in the wrong place. To avoid that, crossing over does not happen really close to wherever the crossing over already happened, which means two genes far apart are more likely to cross over together. And that's why higher recombination frequencies usually indicate longer distances within the chromosome, and therefore you can use that to actually determine the recombination between two genes. Now remember that by independent assortment, the maximum recombination frequency is 50%. So any gene that has a recombination of at least 50% could theoretically be on separate chromosomes, where then that would explain the independent assortment. But if that's not necessarily true, you have to go ahead and look at relationships between other genes to see if that's the case. For example, these two genes could maybe be somewhere close to 50%, but still be in the same chromosome. And the reason you know they're in the same chromosome is because there's a second gene that bridges them, let's say about 25% apart each. So you know that they must all be in the same chromosome in order for this to ever be possible. Because if all these genes were in different chromosomes, the ratio between all of them will always be 50%. So that's kind of how you differentiate between genes in different chromosomes and genes within the same chromosome when you're trying to do linkage mapping. Another type of mapping that has developed much later than this kind of mapping is actually using a way to tag the genes. So when you talk about later, we're going to talk about the double helix. We're going to understand that the structure of the DNA molecule. But basically, there's these primers, or we talked about this again when we talk about biotechnology later in the year. But these primers are molecules which attach themselves to the genetic code and can be seen with the electron microscopes, and the picture will be in color. And so you can find the position of the genes by the different colors that they have. And look how that's actually even more precise than a linkage map would be. Look at that. You can actually see where the genes are within the chromosome because they're tagged by color. Because these probes are primers, or probes are attached to the chromosomes at the DNA level, creating this view of the chromosome. And you can also use this to identify in which chromosome the gene is, because you can see that when you add the probe, only the chromosomes that have the gene will light up in the screen. So you see the presence of the gene in several chromosomes in this picture. So you see that chromosome 2, 9, and so forth have these genes. And so you can see the distribution of a certain gene across the genome. And if this gene is in so many chromosomes, you can think that there's something going on here. This gene must be important, because or maybe it's a part of a gene family that has been transposed to several chromosomes before. But either way, that's how you do cytogenic mapping. And by doing this cytogenic mapping, we've actually identified a lot of genes within the human chromosome and actually found similar genes in other animals as well. So you see, for example, here, a comparison between genes within the humans and the mouse chromosomes. And you can see how much similarity actually exists within that. And look how, by the way, how chromosome 11 and 17 are have a lot of the same similar genes, and how 12 and 10 have a lot of the same similar genes and so forth. Alright? Now, another way of doing mapping, and this is way more complex, it's called fragment arrangement maps, or physical mapping. Okay? Now, basically this happens like this. You sequence small pieces of the DNA. So, that's what you're seeing here on the bottom, on F. So you're going to go ahead and you're going to sequence several sequences of the DNA, and you do this through several different mechanisms, which I'm going to talk about later, and when we do a biotechnology. But after you have the sequence of a fragment, and you get that sequence, and you get the sequence of another fragment, you can see overlaps between one sequence and another. And where those overlaps happen, you know that those genes must connect to each other, and basically you stitch those overlaps together to make a picture of the entire chromosome. So, for example, this chromosome A has multiple genes inside of it, and it's by stitching those genes together and everything that's supposed to be in between them according to the sequencing that you actually create that. And this is called physical mapping. Alright? Another way of mapping is actually called gene sequencing or genome mapping. And by the way, with sequencing mapping, you can combine that with the cytogenic mapping to make a human genome map. So you can actually get that sequencing mapping that you did, that you can see here in the two chromosomes, and combine that with cytogenic mapping, which you see in the bottom, to make a human genome map that we see on the left side. Alright? Now, that is where we were in the 90s, before modern computer technology allowed us to sequence the genome step by step, letter by letter, base by base of the DNA code. Now we've actually gotten to the point that we can actually do other types of sequencing. So, for example, you can get a DNA strand, and base by base, you can sequence it. So, for example, you can tag each of the bases in the DNA code with a radioactive marker. All right. So, for example, you put a blue marker on C, a yellow marker on G, a red marker on A, and a green marker on T, and basically you create these primers which end with those markers. So now then you go ahead and sequence the DNA, and you're going to get different pieces that end with different markers. And by aligning up these different pieces, you actually get the order. So, for example, we know this sequence has to be G, T, A, A, C, G, G, A, T, C, A, T, C, G, because we know the first one was, the last one there had to be G, because that particular piece ended with the marker in blue. But this particular piece ended with the marker in red and yellow and so forth. And we do this in a little more detail when we do biotechnology later in the year. But basically, by looking, this is called the Sanger system of genetic mapping, and by doing this system, we were able to actually look at the actual sequence of the DNA, and then by studying the sequence, you can find out where a gene starts and where a gene ends. So it's a phenomenal way of doing that. And a variation of this is actually combining the idea of physical mapping with the idea of genomic mapping. So getting the actual, you don't necessarily have to code the entire chromosome step by step using that little primer ending in a letter thing, because that would take you forever to do one step at a time to get the entire genetic code. Instead, you can sequence small pieces, line up all the pieces the way we do in physical mapping, and then get the entire chromosome code, or the entire genome that you're interested in doing. And you can actually, a variation of that, of the shotgun sequencing, this is called shotgun sequencing, right? Where you get the code, you split into pieces, you sequence each code, you find overlaps, and then you create the final sequence. So it's a better way, faster way, than if you were to sequence the whole thing, piece by letter by letter. And then you can do that even faster if you sort them by type of chromosome. So if you actually order them, or computerize, you do a computerized assembly that makes logical sense. You know, if you know or where the position is in the genome, say with something like a cytogenic map or something like a linkage map, then you can use that as a guide to actually sort them in the position, which makes the overlapping process even easier. So in other words, if you start combining these different ways of creating the map, you actually can make an even faster method of coding. And then the very most advanced way that we use today is actually called instant mapping, or a process by which you get the DNA, you fragment it, you clone the piece that you want, and you amplify that piece many, many times, and then you do something that's called instant sequencing, where you actually use a probe or a device that measures the consumption of certain chemicals in the solution. And so you put all the bases that the DNA needs to be copied in there, and as the DNA is being amplified and copied by the machine, you can actually calculate use or disuse of certain bases, and then you know the order is. So for example, when you see that the C was used a lot, you know that that is definitely the next part. So you see, every time you see a spike in the C, you know that that's the next one. Every time you see a spike in the T, you know that that's the next one. And so by putting those spikes together, you can instantly get the code for the entire sequence as the DNA is copied. So even as the DNA is copied, you can get that code. So that's an even more advanced way to actually sequence the genome. So it's a very interesting way to actually understand the different kinds of mapping. So you have linkage mapping, which looks at combination frequencies between the genes. You have physical mapping, or cytogenic mapping, which looks at tagging the genes and looking at where they are in the code. Then you have the actual physical mapping, which is overlapping different pieces which are already sequenced, either by cytogenic, genomic, or physical mapping, and then line them up together to create the overall chromosome. Or you can actually look at the base sequence itself, which is called genome mapping. And you have three methods of doing that. You have the Sanger method, which looks at the termination of the sequence with a tagged base, or you can have shotgun sequencing method, when you do the same thing but with different pieces. And there's a variation of that where you actually combine that with other kinds of mapping so that you know what each piece belongs to accelerate the overlapping process. And then you finally have the amplification of the genome, followed by an actual instant sequencing and copying of the DNA. And while the DNA is being copied, you actually detect the consumption of the bases, and you can actually see the actual order that the genes are appearing in the code. When you study biotechnology, we look at all of these things in more detail, but I need you to understand there's several ways of copying the code. And what you need to know at this level is, if I say cytogenic mapping, oh, you know, oh, that's tagging. If I say physical mapping, you know, oh, it's getting the fragments and overlapping them. If I say gene sequencing, it's looking at the actual code of the DNA and putting them in order to find the genes. And if I look at linkage mapping, it has to do with the recombination frequencies that Arthur Stewart Vaughn and Thomas Morgan worked on together. I hope you understand what this is all about. If not, please ask questions, and I'll see you in class.